Welcome to Loop Catalog. The aim of this video is to introduce you to the Loop Catalog website, to demonstrate how to navigate the various pages, and to show you how to utilize our embedded analysis modules. The Loop Catalog is a resource of 1,319 HiChIP samples derived from 750 human and 281 mouse datasets from publicly available studies dating 2016 to January 2024. In addition, the Loop Catalog possesses 44 high-resolution Hi-C samples from the 40-nucleome data portal. The backbone of the Loop Catalog database is high-quality chromatin loop calls from uniformly processed HiChIP and Hi-C samples. We will begin this tutorial on the home page of the Loop Catalog. Users should first select a target reference genome of interest. At this time, we support HG38 for human and MM10 for mouse. Users can then select their analysis of choice: loops to access all chromatin loop calls, SGLs to access our SGL analysis module, or stats to browse Loop Catalog summary statistics. We will begin by choosing loops.
To begin, the navigation bar provides access to all analyses previously mentioned and additional pages not available through the homepage itself. The Loop Catalog text here and here provide links back to the home page. Loops will load the main loop page we have here. Tools contains results from secondary analyses using our HiChIP loop calls, including GWAS and eQTL SGLs, Motifs, and statistics. Docs covers topics including pipelines, science, and facts. And lastly, Teams loads a gallery of the Loop Catalog team. The Loops page contains 4 main sections: the WashU Epigenome Browser Explorer, Loops submenus, filtering menu, and the data table for our HiChIP and Hi-C samples. The WashU Epigenome Browser Explorer is an embedding and lightweight version of the full website. Basic functions such as locus search and track reorganization are available. And simple track modifications such as color and filtering on quantitative values can also be performed. As we will see shortly, Loop Catalog samples and their loops and/or peaks can be loaded into this browser. Next, we jump over to the data table section, which contains all samples with the ability to sort on multiple columns by holding down the Shift key. The data table contains the primary name, study, number of reads, biological replicate, hold down our Hi-C, and the number of loops at 5 KB for FC loops and FH loop configurations. In particular, the number of loops and peaks may be -1, indicating an NA or not associated for that analysis. To browse through our dataset, we provided this filtering submenu. To filter on HiChIP pulldown or Hi-C data, you can click on the title here. Let's say you're interested in HJK27 acetylated HiChIP data. You can click on the checkbox here, followed by the filter button. To reset the table, you can click Reset All. And again, click Filter. To collapse all items, click Collapse All. Returning just above to the 4 loop submenus, This corresponds to our 4 loop types. FC loops are derived from HiChIP data processed with HiChIP and ChIP-seq peaks. FH loops are derived from HiChIP data processed with HiChIP and HiChIP peaks. Hiccups loops are derived from HiChIP data processed with the Hiccups pipeline, and HiC loops are derived from high-resolution HiC samples using Mustache. Within each submenu, you will see a selection of resolutions and peaks when appropriate. By clicking on 10 KB, you can see I've added this column to the table. Similarly, we can do the same with 25 KB and peaks. A toggle button is also present to deselect all or select all in one go. Buttons just above the table have various interactive functions. I will describe these from right to left in order of importance. As you may have noticed, the loop and peak columns contain checkboxes. These can be clicked on for selection and visualized using the Visualize button. Here's an example. This will reload the loops page with a new WASHU epigenome browser rendering. The selected tracks are displayed below the gene and repeat master tracks. Between visualizations, the selected samples and order of the table is maintained. And here we can take a quick glance at our results. To expand the usage of our tracks to the original WASHU epigenome website, we developed the Download WASHU JSON button located here. This will download a JSON hub for further usage. The track files themselves can also be downloaded using the Download Data button. When multiple samples are selected, these will all be stored within a tar file. Next, Clear Browser will remove tracks from the browser. Clear Selection will remove your selections from the data table. Copy will copy the data table to the clipboard. CSV will instead download a comma-separated file of the data table. Lastly, column visibility can be used to show other metadata columns that have been hidden for visualization purposes. From here, we move on to our HiChIP sample pages, which are made available by clicking on one of the primary names within the table. We will use a KYSE 140 cell line sample, which is very data rich with respect to our website functions.
Sample pages begin with metadata information about the sample. This includes study name, organism, chip pulldown, GeoID, GSM IDs, and number of read pairs. Following the metadata, we provide loop call and peak call statistics for FC loops, FH loops, hiccups loops, ChIP-seq peaks, and FH peaks. Please note that links to download peak call and/or loop call bed files are also readily available. For samples belonging to the high confidence regulatory loops, or HC-reg loops sample subset, we scanned for 727 known motifs from the JASPAR database in the ChIP-seq peaks at loop anchors of 5 KB loops using meme suite FEMO. We make these results available as an embedding of the FEMO results HTML page. For those samples containing FC loop data, we also generated enhancer promoter networks with community detection and ranking. We performed an initial round of community detection, and the largest network is visualized here. we performed a second round of community detection, and these we coined subcommunities. In the graph, these subcommunities can be seen as subgraphs with different colors. To investigate other communities and subcommunities, a table is provided below with several filtering options to help with the presentation of the data. To view additional C-rank metrics, click the Other C-rank Metrics button. Filtering can also be performed by sliding any one or multiple of the range bars below, and then selecting Filter. All filters can also be removed by selecting Reset Table. Our recommendation is to search for communities with a high community C-rank score, in addition to a high subcommunity C-rank score. We will next access the SGL analysis module, starting with the GWAS SGLs. Please note that the GWAS SGL module can also be accessed from the home page.
SGL is short for SNP gene pair with a loop, and within the context of the loop catalog refers to the intersection of GWAS, eQTL, and genes with HiChIP data. So why use this tool? We know that approximately 90% of GWAS SNPs fall within non-coding regions, and 3D chromatin conformation can facilitate gene expression via loop formation. Therefore, SGLs can be a powerful mechanism linking SNPs to their target genes, and thus we present our SGL resource using 5 KB FC loops from the loop catalog. There are 2 stages to accessing loop catalog SGLs, including the entry and results page. Given the large number of SGLs, the entry page moderates access to data by 1, disease, of which we currently host rheumatoid arthritis, atopic dermatitis, type 1 diabetes, and psoriasis, with many more to come. Then locus allows gene, variant, and coordinate-based queries, and the sample selection menu currently hosts only immune-based samples matched to the same 4 diseases. For an example, click the example button, then query. The results page is then loaded with 3 main sections: a WashU epigenome browser embedding, locus menu, and SGL data table. The embedding contains the SNP track, as shown here, as well as loop tracks for each of your samples. The locus menu then allows requerying while recycling the same disease and samples. Note, this requires a page reload. The data table down below allows multiple column sorting and the use of copy and CSV buttons as in the loop page. Each row corresponds to an SGL within a single sample. To interrogate an SGL, we design navigation buttons that allow you to jump between the SNP, the left anchor, right anchor, or the loop as a whole by clicking the distance button. In addition, find mapping and loop statistics in the form of FindMap PPA and negative log 10 Q value for loop strength is provided. Next is the eQTL SGLs. Please note, this tool is only available via the navigation bar. eQTL SGLs use a similar representation as GWAS SGLs, with an entry and results page. We will click the example button as before and query. Like the GWAS SGLs, we have a WashU epigenome browser embedding with an eQTL SNP track, locus menu, and data table. Given the use of eQTLs, we also provide the eQTL PIP statistic here in addition to the loop string. Next, we will access the Motifs module using the navigation bar.
This page provides 3 reports from 1D motif enrichment analysis performed on conserved regulatory loop anchor regions from high-confidence HiChIP sample sets. The full high-confidence HiChIP sample set contains 53 H3K27AC HiChIP samples, and the immune and non-immune sample subsets possess 27 and 26 samples respectively. Loops present in 90% or more of the samples in a given sample set were one-dimensionalized, and the motif enrichment analysis was performed using meme suite C. Here you can find the C results HTML pages and accompanying statistics for each analysis. For more information on reported values, click on the question mark at the top of each column. The final tool currently available is statistics. Please note that statistics can also be accessed via the home page. This page provides various statistics on the HiChIP samples in the catalog, including distributions of protein pull-downs, restriction enzymes, number of reads, number of loops, and finally, loop distance. Similar functionalities are available for our mouse MM10 data. To access this data, navigate back to the home page by clicking the loop catalog icon either here or up here. From the home page, we are able to re-enter the loop catalog to browse mouse samples by selecting the MM10 reference genome. Please note that SGLs are currently not available for mouse. Here is a glance at the loops page for mouse. We will now explore the Docs button. Under Docs, we can access pipelines, science, and facts. To begin, let's look at the pipelines page. This page contains technical details for the various pipelines we developed and implemented. Next, the science page provides information on HiChIP's popularity and is where users will be able to access supplementary background information about the HiC and the HiChIP assays. Finally, facts contains answers to some popular and irrelevant questions.
The last navigation button guides you to our team page, where you can find a gallery of the Loop Catalog team. It is thanks to everyone's effort here that we're able to make this website and resource possible. Lastly, the footer contains a link to our preprints and publications. Thank you for taking the time to watch this video. We hope that it was helpful, and we wish you the best in your analysis. Please feel free to contact the Loop Catalog team with any questions, comments, or concerns.